So good evening, everyone. Welcome to Science at the Theater's Extreme Science, sponsored by the Friends of Berkeley Lab. My name is Jeff Miller, Head of Public Affairs at the lab, and your host for the evening. First, I wanted to let you know that X-Science has nothing to do with X-Files, although our presenters tonight certainly have some surprises in store. So I'm going to introduce them individually. They can just tell a little bit about themselves. So we'll begin with Caroline Ajo-Franklin.
Hi, I'm a staff scientist at the Molecular Foundry at Lawrence Berkeley National Lab.
Next, Mr. Andrew Miner.
Hi, I'm a professor of material science and engineering at UC Berkeley and a scientist at the National Center for Electron Microscopy at LBL.
And Tamas Torok.
Good evening. I'm a microbiologist at the Berkeley Lab.
And Spencer Klein.
Hi, I'm a senior scientist in the Nuclear Science Division at Berkeley Lab.
Well, one of the surprises tonight I'm going to reveal now, it's kind of obvious we have this sign that says South Pole. So sometime tonight, we hope that this phone will ring and that it's not a solicitation, that it's actually colleagues of Spencer's from the South Pole, and we're going to have a conversation. We'll see how that works. When we chose the topic extreme science, we did so because we wanted you to understand that our scientists don't kind of sit in the lab and magically hope that discoveries will appear. They actually go to extreme lengths and travel extreme distances and sometimes do their work in extreme environments. The results are, well, kind of extremely interesting, but I'll let you be the judge of that. Okay, we're going to begin the presentation again. The phone is scheduled to go off at, we think, at 8 o'clock, but it might come earlier, so if it does, we're just going to stop and answer it. But I will begin with Mr. Miner. Please welcome him.
Thank you. It's really a pleasure to be here tonight. And so what I wanted to talk to you about tonight is materials in extreme environments. And these environments can be, for instance, high temperatures, like the nickel superalloys that are inside a jet turbine engine. They have to operate at 1,500 degrees Celsius or higher. It can be environments like inside nuclear reactors where the materials are containing the reactions are continually bombarded by neutrons and protons. It can be extreme strains, like when you're forming materials to form cars, for instance. And two things I wanted to point out on this slide are that, number one, up top, performance limits really result from insurmountable physical principles. In fact, most of the engineering materials that we have actually only operate about 5 or 10% of the theoretical strength. So there's a lot of room to grow. There's a lot of room to make better materials that can handle these extreme environments better. And lastly, one thing that would be really nice is in these extreme environments to, for instance, be able to test the samples that have been in these environments or while they're in the environments, right? You'd love to take a little sample from inside this nuclear reactor and be able to test and make sure that it's still okay and that it'll last as long as you want it to last. But it's a little bit harder than you might expect. So the other thing I wanted to point out before I show you some of the results is that a lot of you might not know is that materials that are useful are typically crystalline. That means that the atoms inside the material are all lined up in rows, like something like the pictures at the bottom here. In addition, in fact, one of the interesting things is that the only material we actually call crystal turns out to not be a crystal. It's actually amorphous. But most of them are crystalline, and more than that, most of them have lots of defects inside them. That means that, for instance, in the bottom left here, you can see there's a missing row of atoms. And this is what's really important about crystalline materials. For instance, these missing rows are called dislocations. And the fact that these dislocations can move easily around inside a material, that's what makes metals ductile. So understanding these defects is really important to understanding the performance and the properties of materials. And there's a great quote, of course, here from Sir Frank that says, "Crystals are like people. It's the defects in them that make them interesting." And that's really true. So here's the question. So how do you see dislocations? Because they're very small. They're single rows of atoms. Well, of course, you need a good microscope, right? And so if you look at the difference between a caveman microscope, as drawn here by Gary Larson, and a light microscope, there's many orders of magnitude difference in the ultimate resolution, what you can see. Well, the difference between a light microscope and an electron microscope is also many orders of magnitude, 4 orders of magnitude. And by using electrons instead of light, in much the same way, except for one small difference, and that's that you actually shoot electrons through the material so you can see inside it. You can actually see down to the atomic level. And the lab that I work at LBL is actually called the National Center for Electron Microscopy. We're lucky to have some of the world's best microscopes. In fact, these are two of these microscopes shown down here that each have half an angstrom resolution, which is about half the width of one atom. And these are both, let's say, tied for the best microscopes in the world. And for instance, it takes pictures like this. You can see this is a gold nanoparticle, and all these little white dots, those are atoms. And you can see where the atoms are crystalline in the middle, and they're all lined up nicely together. And around the side, they're not. They're kind of squiggly, and that's the surfactant, the organic molecules on the outside. So these are the kind of pictures you can take, and you can look inside and see defects and see material structure. Okay, that's great. So we can see material structure. We can see these dislocations. But how do you make test samples from these extreme environments? Well, it turns out there's this great machine called a focused ion beam. It's really amazing. You can actually mill samples down to the nanoscale, machine them just like you would with a big lathe. But instead of using drills and things like that, you use individual atoms. It's kind of like a sandblaster, but you focus the atoms down to a small point and you mill out different structures. Okay, so you can take materials and you can mill out little test samples and you can put them in a microscope and see inside them. That all sounds great, and you can do tests like this. You can take little pillars like this. This is a pillar of nickel, and here it's been compressed, and you can measure the strength at which this material deforms. That's great, except there's a rub, and the rub is that the strength of materials actually depends on the size. That's not totally intuitive, right? If you look at Wikipedia and you look at what's the strength of nickel, you're going to get a number, and it's 200 MPa or something like that. Well, turns out that's true for almost any material you can handle with your hands. But actually, as you get smaller and smaller, microscopic and down to the nanoscale, the properties themselves change. And that's because of having a lot more surface area and actually the spacing of these defects and things like that. So the problem is shown here in this graph is that this is about this dashed line is the macroscopic strength. That's what you measure if you had a bar of nickel and you just bent it. But as you go smaller and smaller, the strength increases. So you can't just sort of do one of these nanoscale tests and say, okay, now I know this material is still good. You have to understand how strength changes at these nanoscale levels. So this is the type of tests we do at NSM in my research group. We do small compression and tension and bending tests. And for instance, here's an example here on the left. This is what it looks like in an electron microscope, this big circle here. You can see there's some pillars. Here's one that has not been deformed. Here's one that's sheared off to one side. Here's one that's sheared off to the other side. And there's this big flat punch indenter right here. And I'll show you a movie of what this looks like right here. So as we're compressing, you can see a bunch of these little lines move around. The scale, for instance, you see this red line up here? That's about the width of a red blood cell. And so we're doing tests at that small scale. And the diameter of these pillars, as you can see, they're about 200 nanometers, okay? So that's about 10,000 atoms across. And here's another one here. See, this one works a little better. You see these lines that go back and forth at the bottom here? Well, these are actually dislocations. So every one of those lines is a row of atoms that moved on another row of atoms and let this material deform. So now we're actually seeing inside a material how the defect structure evolves. So we're measuring small scale samples, and actually at the small scale we not only measure the properties, but we actually can also see new mechanisms of deformation that might lead to better alloys and better designs. So for instance, this was a project I worked on with General Motors, and they're very interested in making cars out of magnesium because magnesium is very light. So you'd love to make a car out of magnesium. It would have great fuel efficiency. So here we take little dog bone samples. These are, instead of a pillar that we compress, you can actually take, for instance, a little sample and pull it. That's called a tension test, right? So here's a little dog bone, and we have a little gripper. We can come in and we can pull it. And you can see in the bottom left here, this is the sample breaking. And in the middle bottom right here, you can see there's a bunch of these triangular-like streaks. And these are the defects that were generated during this test. And it turns out these defects are something different than a dislocation. They're called a twin. And a twin is like, if you look at this animation at the top right, the materials deform by flipping over one another along a twin plane. And this is actually an atomic resolution image at the bottom right here of these small nanoscale twins inside the sample. And these twins actually are so small that they give you both great strength and ductility. So if we could, for instance, figure out how to make a material that generated these twins in the bulk scale, then we'd actually have a much stronger material that we could form into the type of parts we needed to form them into. Now, as far as extreme environments, testing materials in these extreme environments, small-scale testing has a big advantage there too. So for instance, this is a material that had been irradiated with protons. This is simulating what would happen inside a nuclear reactor. Because for instance, you know that it would be terrific to be able to take a little slice off the inside of a reactor and be able to tell that this material has not turned brittle over time like they usually do from the bombardment of neutrons, but instead this material is still ductile and it's still damage tolerant and all these things. So you would love to be able to do a small-scale test of a irradiated sample. And what we found here was very interesting because, like I said before, you always have this general trend, smaller is stronger, and because of that, you really can't get a real value out of these tests. Well, it turns out in these irradiated samples you can. And the reason is because there's so many defects in these materials, and these defects, this is a little, this picture here in this yellow box is you can kind of see a triangle-like shape in this atomic resolution image. These are called stacking fault tetrahedra, and this is the type of defects that get put into a material inside a reactor. Well, it turns out there's so many of these defects that the strength of this material is actually dependent on the spacing between the defects and not the actual spacing of the sample. And that's what this graph shows here, is that you can see here's this general smaller, stronger trend, but for the irradiated samples, the red dots, about 400 nanometers, you start to get a plateau. You start to get a plateau. And what that tells you is you're starting to get this macroscopic shear strength. And so really what you're testing is the real properties of this material, even though it's so small. And so these types of developments that we work on, whether it's testing or new materials, it's all about trying to understand the defects, how they evolve, and how the material's properties change with time so we could design better materials for applications like this. So with that, I'll let Caroline take over. Okay.
Okay. All right. So good evening, and thank you all for coming. So what I'd like to tell you a little bit about today is really my own personal fascination with living systems that survive in extreme environments, and also how we're trying to harness the unique capabilities of these living systems to actually benefit society. So one of the things that's really amazing about life is that throughout Earth's history, you can find life, particularly microorganisms, that thrive in extreme environments way too harsh for us delicate humans. So for example, in the earliest days of Earth's history, there was life began as microorganisms in an environment that was pretty much completely free of oxygen. And even today, you can go to deep sea thermal vents or to sulfur vents on the side of volcanoes, and what you'll find is teeming communities of microorganisms in environments that really first are too hot for us and also are the wrong atmosphere. So we would basically suffocate and cook in these types of environments. But life survives. So how do these organisms how can they survive in environments that are free from oxygen? Well, it really comes down to taking a deeper understanding of why we need to breathe oxygen. So in the cells of our bodies, what we do is our cells are designed to basically take food, for example, sugar, and break it down and actually move electrons from that food to oxygen. And that chemical reaction, that passing of electrons, is what gives us energy to move, to talk, to walk, to gesticulate, or stumble over our words, whichever. So for these organisms that have evolved in oxygen-free environments, they've learned to breathe something completely different. So what they have learned to do is actually to breathe metals. So that they can actually take the electrons from their food and pass it to the metals. And that's how they get energy to go ahead and divide and grow. So, and you can actually, using simple microscopes, not the fancy ones that Andy has, you can actually watch this happen. So let me go ahead and show a little movie. So what I'm going to show you is a movie in which there are hundreds of microbes that actually surround this rock, this piece of metal-containing mineral. And you'll see that it slowly starts to dissolve as the microbes actually move electrons to this material, because basically that reduction of the metals causes it to dissolve. Okay. So what you see is here's the rock, and it slowly starts to dissolve. And just to prove to you that this is indeed microbes doing the business and not just me doing some fancy chemistry, I'll show you a zoom in of this same reaction. So here you see the microbes all swarming towards the mineral, because they need this mineral to be able to breathe and to grow and divide. So that's really exciting to me, because basically these microbes are able to move electrons to metals, to wires. And so that also means that they can, and they do this from sugar. So they actually have the capability to create electricity from sugar. And here's a little device that I'll show you in which these microbes are actually powering a little fan. So this is a microbial fuel cell that uses the microbes to catalyze movement of electrons from sugar into electricity and actually fuel a little fan. So this gets me really excited, because I've just shown you that that these particularly metal-reducing bacteria are able to create electricity. So that gets me, as a scientist, really excited, because I'm all too familiar with these sorts of adapters. We all have like a bag full of them, where they're AC to DC adapters or different voltages or for different countries. Well, what it got me thinking about is, could we actually take the ability from these native extreme microorganisms to create a bio-to-electro converter, where we could essentially move electrons or electricity into and out of any organism that we want. So how could we actually take that sort of very specialized trait from these extreme organisms and put it into any organism? Well, the reality is that scientists have gotten really good at this over the last 30 years. And perhaps one of the most famous examples is here, shown on this slide. So DNA essentially carries information, sequences of DNA carry information, and all organisms share the same language of DNA. So what scientists have found is that, for example, if you can take the right segment of DNA from this jellyfish that can glow green and put it into a normal fruit fly, this fruit fly, you can basically confer upon it the ability to now glow green. Now, that's not particularly exciting, for example, for the fruit fly, but it actually has been used extensively to help us learn about the molecular processes within this fruit fly. But basically, this is a trick that we can use for many different types of traits. So we wanted to see if we could use it to be able to create this converter. So what we did is we went to this native organism. We found the DNA that we thought was responsible for the ability to move electrons to metals, and then put it into E. coli. E. coli is just a laboratory organism. It's a little bacteria that you can find tons of in your guts. But scientists have been using it extensively as a way of studying bacteria. And native E. coli can't reduce metals at all. So when we took this DNA and put it into E. coli and wanted to ask the question, could we make electric E. coli? or E. coli. And so what we found when we did this was that the bacteria, our new E. coli, like to actually bind to metals. And then it got even more interesting, because what we did is we took E. coli, the normal strain, gave it sugar, and then removed all the oxygen, but added in some metal. And normal E. coli says, this isn't helping me at all. I have nothing to breathe. I'm just going to die. Whereas our electric E. coli basically can survive for a little, because now it can breathe the metal that we've supplied to it before it also tanks out and dies. So that's really exciting, because that basically tells us that we're able to give an organism the ability to breathe metals instead of oxygen. What's even more exciting is that we can now actually get electricity from this bacteria. So what we did is fed this electric E. coli sugar, and we can actually measure current that it produces for a while, and then it slowly decays. But then as soon as you pop in some new sugar, more food, up comes the current again. So this is really exciting to me, because this is actually evidence that we have this bio to electrico sort of converter. And it got my imagination really going about what can we do with this. And there are two things that I think keep me up at night dreaming about. The first is the idea of actually trying to create more efficient ways of creating bioenergy. So right now, there are many efforts to take biomass, convert it into sugars, and then have microorganisms convert those sugars into fuels. But one of the big problems is this requires a lot of land and a lot of water. So we thought about, well, what if instead you could take CO2, so carbon, and then a source of electricity, feed it to these microbes, and get them to make biofuels? That would be really advantageous, because essentially it would require a lot less land, and in theory it could use any source of electricity to store that energy in the form of fuels. The other area that we got really excited about is the idea of being able to actually repair sight. So currently there are actually people who have lost their sight who now have these little microchips implanted in the back of their retina. And what this microchip allows you to do is to send signals from a camera to someone's eye, and that your eye can then read those signals and actually give you a sense of sight. So people who have lost their sight now, through this technology, can regain it for a while, because after about a year and a half, essentially this microchip shorts out. So one of the things we're really interested in doing is asking the question, can our genes essentially make this interface a lot more biocompatible, and thus hopefully more permanently allow people to regain sight? So I just want to leave you with the idea that we can take these extreme capabilities from these organisms, isolate DNA that has very unique capabilities, confers unique capabilities to microbes or to other organisms, and hopefully we hope that those can actually impact society very positively by helping with energy independence and, of course, helping with human health. Thank you.
Good evening. Thank you for having me here tonight. We're going to be talking a little bit more about microorganisms. Carl Woese, one of the most influential scientists of our time, in 2002 wrote that where there is life, there are microorganisms. I think this is a very basic statement, but my actual inspiration is a little bit older than that, because we just celebrated recently his 256th or so birthday. No, that's not true. There's a reason I'm a microbiologist, not a mathematician. Anyway, so we just celebrated his birthday, Charles Darwin, who in 1845, some 170 years ago, almost 170 years ago, basically stated that we'll see will find in any possible habitat on our planet some organic material, some life. And he wrote that when people couldn't fly yet to collect atmospheric sample and see whether there are microorganisms in there, there was no Alvin, which would go down to the bottom of the deep sea. There were no helicopters. I will show you pictures, which I took myself from volcanoes. And he already believed in it, that life can inhabit, colonize everything. And as we heard already, that most likely will be microorganisms. So what is more magnificent than Mother Nature at its best? And if you have a chance to study that, that's probably the best you can get, the best as it can get. So my journey basically started a long time ago. But in the past about 20 years, I mostly dedicated my science to bioprospecting. Basically a process where you go out into Mother Nature, because Mother Nature has it all. You just have to look for it, and microbiologists usually find whatever they are looking for. So you just have to look properly. And we'll find old or novel organisms, most likely with some novel genetic traits, which can be very important, because it will advance science, benefit humankind, and at the same time also longer term may preserve the gene pool which is out there. So my story is actually the beauty and the beast. The beauty being microbial evolution or biological evolution, and the beast because about 15, 16 years ago I became involved or became part of a program which Congress mandated and the US Department of Energy is performing, basically engaging former Soviet weapons scientists in peaceful projects, peaceful research, retraining them kind of, and introducing them into the International Society of Scientists. And what is more peaceful than in a huge country like the former Soviet Union, go and bioprospect in areas which were totally untapped in the past. So I was looking for extreme environments to find extremophilic microorganisms which basically cannot survive most of the time under normal, what we call normal conditions, because they are used to, they evolved to be able to inhabit environments which are either too hot or too cold or has unique chemistry or very little nutrients or very radioactivity contaminated. So something is unique about the environment. What you see here, this is an image I took in Kamchatka. We'll talk about it later. And at that environment, water boils at 94 degrees C. Don't ask me what it is in F because I have no idea. But it's 94 degrees C. And I doubt it you can read it, but the thermometer shows 97 degrees C, which means that in that environment, this is superheated water environment, and microorganisms make a living in there. Actually, microbes pulled out from this hole were in the meantime introduced, and they are producing a unique enzyme which allows the paper pulp industry to reduce the amount of bleach which they use to whiten the material by about 30–40%, which is a big deal. Whether you take the money or the environmental impact of using bleach. So once we collect all this material in the environment, it comes to our laboratory, and then in the lab, we'll use a nice combination of culture-based and culture-independent, more novel techniques. And I'll be happy to talk about it if you have any questions about that part. So my first really interesting project took me to Lake Baikal. I don't know how many of you know Lake Baikal, but Lake Baikal sits in the middle of Central Asia, middle of Siberia, basically. And this is the largest lake in the world. I know the Canadians don't like this idea, but it's still fact. As a matter of fact, it is. And it holds 25% of all terrestrial freshwater in just one lake. The average lake depth is 1 mile. And I learned when I was there that there is an international Baikal drilling project going on. And luckily for me, there was no microbiologist participating in the program. So I stayed for 2 seasons with the program and collected core samples aseptically for microbiology research every 2 meters as they came out of the ground. So that was pretty fantastic, and looked then for microorganisms and novel capabilities which might have been hidden from what we called ancient microorganisms. Might have been hidden from the view, because our samples, our microbiological samples, were the first non-coastal samples ever collected in Lake Baikal. So I was lucky enough to be there in the summertime and move around with a boat and collect water samples at different depths. But also in the wintertime, when outside is -38, it's totally frozen. That's what you need, because the drilling barge has to be positioned in place. And then the drilling is going on, and 24/7, the cores are coming out of the ground, and you take samples. And of course, the geologists and paleoclimatologists will say that these sediment samples represent 10, 12 million years of records. But as a microbiologist, I would say probably the organisms in that sample are not that old, because in the pore water, there might be some exchange with the water column above. But nonetheless, the results ultimately showed, and we collected about 2,500 different organisms out of the samples, that these organisms have been in isolation for a longer time. We found features which were not traits, capabilities, genetic capabilities, which were not reported earlier for other terrestrial organisms. I don't know how many of you, in the front row, I see mostly young people, so you won't remember, but in 1986, in April, it's going to be 26 years soon, in Ukraine, a nuclear power plant, one block of a nuclear power plant exploded and released probably the largest radioactive contamination known to humankind in that context. The picture is not very good, but on the left panel, but you might be able to see the destroyed Block 4 building, totally exploded, totally destroyed. On the right panel, what you see is 2 weeks later, the plume of the 131 iodine as it spreads out over the northern hemisphere. So it was pretty horrible experiment, horrible event. For an experimental scientist, this is an opportunity, and a very unfortunate, very tragic accident, but it's an opportunity not even once, hopefully not even once of a lifetime. So what we did in the following 18, 20 years, I worked with colleagues who collected plant and soil material and microbiological material directly from the exploded block, from the surrounding, and then later on there was a so-called 30-kilometer radius exclusion zone where there has been no human activity ever since 1986. And out of that, a lot of plant material was collected and then generated. And right now we are screening these plant materials for unique antivirals. It's a unique application, but we know that plants, especially if you tweak the plants a little bit in culture, they might produce some interesting natural products, and we are screening these natural products if there are any antivirals among them. Closer to my field, about over 2,000 fungal organisms were collected out of the Chernobyl exclusion zone and the exploded block over a period of 18 years. And there are several very interesting aspects to it, but let me just give you two. One is that it gave me the opportunity to collect fungal material in varying distance from the radioactive source and find sometimes strains of the very same species very differently adapting to the radiation, to the contamination in the environment with time and with distance. And you see the features, what made all those fungi capable of surviving in the most radioactive environment, which capability is then missing or is being diluted out as the organism is further found from the radiation source. Another interesting aspect was that, which was news to us, that we know microbiologists, we know that microorganisms constantly monitor their environment and interact with it. But it was news to us that fungal organisms, which were totally differently described so far, can sense radioactivity and then basically grow into the direction of the radioactive source. And you can play that in the laboratory also with small radioactive particles. And actually, when they reach the radioactive source, they grow much better and faster. So far, fungal organisms were considered organisms which need organic material to grow on. That's what their carbon and energy source is. And if that can be proven that this is, and the whole mechanism for that can be found, how they sense the radioactivity and actually take advantage of it as an energy source, that would mean that pretty much all the textbooks have to be rewritten, which is not, this is how science evolves. So we call this unique feature positive radiotropism. The past 7 years, I'm really humbled to say that, but I had the opportunity and the honor to take expeditions to the Kamchatka Peninsula. Kamchatka Peninsula is in the Russian Far East at the Pacific Ocean. This is how far you can go in a country with 11 timelines. And that peninsula basically provides pristine environments because it's the size of California, a little bigger than California. 250,000 people live there, and there are about 50 miles of paved roads. The rest of the traffic has to be done by helicopters. So in this pristine environment, you can find permafrost areas and volcanoes. Basically, this is where the circle of fire ends. And hundreds and thousands of geysers and fumaroles and mud pools and very interesting geothermal and hydrothermal environments. Actually, because one of the major collection sites where we worked for many seasons is about 2 hours by helicopter. So we managed to build, out of your taxpayer, thank you very much, out of your taxpayer money, a little field station where we can work and stay. I don't know if the pointer works, but there's a little dot here on the Google map. That's where our house is, this here. So you can find it on the Google map. And that allows us to spend the time from end of May until middle of September, this is the period where the helicopter really safely can fly. And we collected all kinds of material, always rationally designing what we want to collect this year. And here I'm showing you some results. One focus area was trying to find natural products which these fungal organisms or any microorganism is producing that can be used in crop protection. And we were very successful of pulling out of this mud pool here. It's not wanting to show, so this here on the left plan. A bunch of microorganisms which produce antifungal molecules, which we tested, and the tests are shown on the right panel, and then reverse engineered the whole story and found in the microorganisms the genetic coding for all these peptides or polypeptides. And then, you might not like it, but that's the future, I'm sorry. Put that into plants, crop plants, which now are producing their own antifungal molecules. So the plant pathogenic fungi cannot damage and attack the plant. Now, the options currently would be synthetic chemicals, which are not natural, sprayed all over the plants, or the plant growing, excuse me, with its own antifungal molecules. I think that's a much better solution because it's more natural. How that works, a couple of pictures that plant cells are kind of harder to work with. So what you need to introduce microbial genetic material, new genetic material into the plant cell, you use a so-called gene gun, which uses high pressure to blast into the plant cells the new genetic material. And on the right-hand panels, you see some examples how that then looks like. Another area of interest was finding insecticidal molecules. Again, Mother Nature has it all. Microorganisms and insects have been interacting with each other for billions of years or millions of years. So what we were looking for, there are certain bacteria which produce some crystalline protein material which acts as a natural insecticidal molecule. So I don't know if you can see it, but in there are big blocks, nice blocks of protein material, which when the insect is chomping on the plant material or tries to, and the plant material has these bacteria on its surface, then those crystals will ultimately poke holes into the guts of the insect, and the insect will die. Now again, the story goes that we can produce in the laboratory these clusters of crystals. That's not a big deal. But the big deal is to try to find the genetic information for the production of these toxic molecules and put that into the crop plant and let the transgenic plant do their own protection. Lately, we expanded bioprospecting into new areas with new foci. We are looking for organisms which can survive in very salty environments with very little water. Water will be a major issue in our future, and especially in the younger generation future. So we will need plants which can do it without, or make it without with much less water than what we need to have today. So microorganisms again can help. The gene pool is there. We just have to find the proper ones. We are working in Azerbaijan. Azerbaijan has a lot of oil contaminated environments, and we are isolating organisms which can degrade oil contaminants, because oil contamination happens, as we know, sometimes here in the Gulf of Mexico and so. And the latest focus was finding really efficient organisms which can degrade lignocellulosic material. You just heard about the option of making biofuels. And there are many organisms which can do that, but none of them are so successful like these kind of fungi. And I have here 4 different kinds for you to enjoy the image, which grow on either live or dead woody material. These are the most successful. And we have a whole screening program for finding these organisms in different versions of trying to produce these enzymes. And once we have the genetic information which is responsible for the enzyme production that can biodegrade, break down the lignocellulosic material, we kind of speed up the evolutionary process in the laboratory and play a little bit with the DNA molecules. We already heard the term DNA molecules. Play a little bit with the DNA molecules and actually make much better enzymes than Mother Nature at this time of the evolution, this point of the evolution, really managed. We have been successful. We have reported much better enzyme productions for this unique complex called lignocellulose than anybody else so far published in the world. So we have a lot of patents. This is how you measure success when you work with the industry. Now and then you're lucky and you can publish something, but most of the time you measure success by patent protection for your intellectual property. So to sum it up, what I have to say, it humbles me simply to be able to go to all these extreme environments, whereas we heard no other creature could survive but microorganisms. What you see here is a crater lake with a pH of 0.75. That's pretty acidic. And And microorganisms make a living in there. We collected microorganisms from that crater lake in Kamchatka. So these bioprospecting expeditions allowed my laboratory and myself to improve tremendously our culturing techniques, our molecular level techniques, and especially scale-up protocols, because whatever it happens really successfully in the laboratory, it's not a given that it's going to happen in larger industrial scale. Actually, it's still more an art than a science. But what we have done is also of incredible value to the industry, because with my partners, with our partners, it provided them the cutting edge to generate transgenic plants, and these plants spent several years in a greenhouse and now are growing in a field, in an open field. We have a couple of other ideas in a drawer. This is how you try to build a future on post-harvest modification, edible vaccines, not with a needle, but you eat it. And of course, lignocellulose biodegradation. And again, as I said at the beginning, I'm very proud that I took this picture of course, from the helicopter, but from a volcano, because in Kamchatka, there are always about 20, 30 volcanoes active. Thank you very much for your interest.
Let's see. So now for a little bit of a change of pace, I'm going to tell you about some extreme astrophysics. The physics I do is to try and look for some of the most extreme environments in the cosmos. And this shows you an example of one. This is a galaxy, this galaxy. The center, it's got a supermassive black hole, which is rotating, which gives it a bit of a magnetic field. And you can see that there's a jet of particles coming out perpendicular to the axis. That jet is moving at relativistic velocities close to the speed of light, and we believe that it's possible this may be the accelerators that produce the ultra-high energy cosmic rays that we observe on Earth. To study these jets, we use a rather special tool called a neutrino. Neutrinos are interesting because they go through almost anything. Most of the things you know, light or alpha particles, are stopped by a piece of paper. Other types of radiation, beta rays, are stopped by a few centimeters of aluminum, and you can even stop gamma rays or X-rays with a few inches of lead. However, neutrinos will go through almost anything. And by that I mean you can have neutrinos produced even in the center of the star, for example, our own sun, and those neutrinos will make it out of the sun, or out of that star. They can travel, they can enter the Earth, they can go through the Earth, and occasionally one of them will interact in Antarctica and produce a shower of particles coming upward out of the Earth. And that's what we try and study. We do that with two detectors that I'll tell you a little bit about in a second. The one thing to keep in mind is that because neutrinos go through almost anything, that means they don't interact very much. So that means you need truly enormous detectors to be able to see them. So we use two detectors. The first one is called IceCube. It's located at the South Pole, well, actually about a kilometer away, but at Amundsen Scott Station. And that's a detector that is up and running, and I'll talk about that a bit. And then we're also working on an experiment called Ariana, which is on the Ross Ice Shelf about 100 kilometers south of McMurdo Station. And you can see here in black, McMurdo Station. This is the main US logistics base in Antarctica. Excuse me. So Ariana, in contrast to IceCube, is just getting started. We deployed a prototype station there a few years ago. So the other thing to keep in mind about working in Antarctica is it's a very difficult environment. And things that are would be very simple in a lab at Cal or at LBL can be very difficult. You know, even things like providing power, that's an enormous struggle. And of course, things have to be extremely reliable. And the big thing is you need everything you need, and you need it there. You can't just run out to a hardware store. So an awful lot of what I'll talk about, there's an enormous effort in logistics, considerably more so than actually doing the science. And I'll show you some examples of that. So to get to Antarctica, you start out in Christchurch, New Zealand. And of course, there's no regular flights to Antarctica. So the Air Force provides planes. This is a C-17 landing literally on the Ross Ice Shelf. You get off, and well, this shows the inside of the plane. You can see passengers on the side, cargo in the middle. That's so the passengers are there to protect the cargo if anything shifts during flight. So then and in McMurdo, they have a lot of specialized heavy vehicles to be able to move people around and things around. If you're going on to the South Pole, you will then shift gears and shift planes and fly on one of these smaller LC-130s, actually operated by the New York Air National Guard, despite what it says on the side. The notable thing here is you can see this plane is landing on skis rather than on wheels. And that's because of the snow conditions. So, and once you get there, then you can start working. This shows an aerial view of the South Pole. You can see up in blue is the new, I don't know, this is, okay, well, maybe you can see, this shows the new station. The actual South Pole is here. This is the skiway. And then the experiment ice cube is kind of located all around here. There's not much to see on the surface. This counting house contains our surface electronics. And this drill camp is something we use for constructing the experiment. But most of the action is under the surface into this bottom kilometer of ice. What we do is we drilled 86 holes down, each 2,400 meters. It's about 1.5 miles. And then we lower a cable, like that shown here, into the hole with these optical modules. There's one there. And this is a prototype optical module. You can get an idea what it looks like. It's made of 1/2-inch thick glass on the outside. And the business end is a phototube, which is sensitive to light. And so what happens is occasionally a neutrino will come in and interact in the ice. It will produce a bunch of electrically charged particles. This could be electrons, positrons, or other particles. These particles will move through the ice near the speed of light. And as they do that, they will emit light, known as Cherenkov radiation. And by looking at the arrival times and amount of light in the different optical detectors, we can get a pretty good idea of the neutrino direction, often to within a degree, and some idea of the energy. So this shows the main activity in IceCube, which is actually a very mundane construction activity, drilling the holes. I showed you this drill camp here is essentially a 5 megawatt hot water heater. It uses technology that's borrowed from car washes, and we use that to produce 5,000 gallons a minute of water that's heated to 88 degrees centigrade. And then that water comes and goes through here, through this drill tower, and we just melt our way down through the ice. It takes about 40 hours to get down to 2,400 meters, and then about another 12 hours to deploy the cable and the optical modules. And again, getting back to the logistics, I just like this picture. This shows the old South Pole Station. It was built in 1975. It was on the surface then. Now, I took this picture in 2006. It's almost buried, and in fact, it has since been disassembled. This shows you the inside under that dome. There's buildings, and typically about 30 people would spend the winter there. Now, we have the new station, and modern technology permitting, we will hear from two of our winter overs. These are people who spend the winter there specifically to maintain the IceCube equipment on the surface and keep everything operating. So Ariana, in contrast, is a greenfield site, or maybe I should say a white field site. So until we went there, there had been nobody at this area, at least as far as we know. So to get there, we go out in a helicopter. This shows leaving McMurdo Station. And then this shows, actually this was a later helicopter arrival at the site. You can see there's absolutely nothing there. You know, when we were flying out there, the helicopter pilot asked where we wanted to land, and we said, you know, anywhere around here. So it dropped us off and then came back the next day. You can see this is our scientific equipment, and these are some of the large tents. And again, this is the point, we had to spend more time, way more time, gathering equipment than we actually spent out at the site. And we were out at the site for 11 days. So our main purpose was to install a prototype detector. This works on a principle that's very similar to IceCube. It looks for this Cherenkov radiation from neutrino interactions, except it's looking for more energetic neutrinos, and that turns out to be better done with radio waves, which are also electromagnetic, just like photons, but much longer wavelength. So the business end of the detector the prototype were 4 antennas like these. Those of you who are old enough will remember traditional television antennas. These are very, very similar to those. And then there's a box, electronics box, that looks at the antennas and looks for short pulses of electromagnetic radiation or radio waves that you would get from a neutrino interaction. And again, that's something that would be quite simple doing it around here. The difficulty, of course, is all the logistics. So in addition to the antenna, we have a box that contains the data acquisition electronics, and then this tower, which contains power. You can see there's 4 solar cells. That works well in the summer. Of course, it's useless in the winter when it's dark for about 5.5 months. There's a wind generator here, which we were hoping would provide enough power for in the winter. It turned out not to. It's not a very windy site. And then there's GPS antennas, so we can know what time it is, and a Iridium satellite modem for short-term communication, or for low bandwidth communication, I should say. I just want to show you, this picture shows Torsten Stetzelberger working in our laboratory. So he was an engineer that went with me. This shows the entire camp. Get an idea of the ratio of logistics to other things. This is our toilet tent. These 3 tents were sleeping tents. You can see the station. Behind it was our cooking and dining and some laboratory tent. And then our equipment box. And then this was the internet link. And this internet link is important because we're only there for 11 days. We eventually want the station to run year-round. We want to be able to talk to it. We want to be able to get substantial amounts of data out of it. Of course, around here, just find the nearest Wi-Fi node. We didn't have that. So, in fact, we had a bit of a problem because this is something called Mina Bluff. It stood between the site and McMurdo Station. It's, I think, somewhere around 1,000 feet high, and it's high enough it blocks radio waves. And in a way, that's good because remember, we're burying these antennas in the ice. We don't want a lot of man-made interference. On the other hand, when you're trying to get your data out on a very different frequency, it's a problem. So what we had to do was went up to Mount Discovery, and Mina Bluff is kind of off to the right here. And roughly where that red dot is, we set up an internet relay station, which had line of sight to our camp and also line of sight to McMurdo Station. And that ended up working pretty well, although, of course, we had to spend some time screwing with it to get it to work. Before I conclude, I just want to point out, an important thing. You may see us as individual scientists here, but science is really, really a group effort, and that's especially true at the South Pole. A huge number of scientists, engineers, technicians, programmers, drillers, heavy equipment operators, pilots. No reindeer, unfortunately. Pilots, logistics, cooks. This is excellent timing, so I hopefully. Hello?
Hello?
Hi, is that Sven?
Yes, hello, it's Sven and Carlos.
Sven and Carlos, hi. This is great timing. We're here at the Berkeley Rep Theatre, and there's people here who would like to know what it's like at the South Pole.
Okay. Yeah, so right now it's minus 49 feet or 54 Fahrenheit. We have a 20 knot wind, so our wind chill right now is minus 70 feet or minus 93 F.
You guys get that?
The sun is right now 9 degrees above horizon, and it will set on March 21st.
Yes, he said it was minus 49 degrees centigrade with wind chill minus 93 Fahrenheit. What are your days like? I understand the station closed last week. How many of you are there for the winter?
We're 50 people here right now for the winter, so we'll be stuck here for 8 months.
Mm-hmm. And what's your typical day like?
It varies quite a bit. I mean, we constantly check the detector, and we do special runs when required. We help out with things on station, and like just an hour ago, we had a fire alarm we had to respond to. No day is like another one.
Yeah, there's people. Hold on. Somebody have a question?
How much time total does this person spend at the South Pole?
How much time have you spent at the South Pole so far? And I guess how much longer do you have?
Okay, so both me and Carlos arrived here on November 3rd, and we're scheduled to leave, and that was last year, November 3rd last year, and we're scheduled to leave mid-November this year. So we'll pretty much be here in over a year.
Okay.
I cannot wrap my head around minus 93 degrees Fahrenheit.
Yes.
I can't wrap my head around that. I don't know what that means. Can you even go out? Can human beings go outside?
So the question was that somebody's having trouble understanding what minus 93 degrees Fahrenheit is like. I mean, how do you handle it? Can you even go outside? What do you wear?
Yeah, so we wear multiple layers of clothes. And wind chill is the hard part. So right today, it's like 20 knot wind. It's actually very cold just because the wind gets through your clothing. But we have big parkas, we have special shoes, and then we just have thermal underwear and just have layer and layer. And try to avoid being outside as much as possible. But, I mean, we also have to take advantage of it. It's only another couple of weeks to have the sun up. We need to do all the work that we can while the sun is up.
Okay. Maybe we take one more question.
How do you divide kind of social versus work, and how much time spent today? What do you do?
Okay. So the question was about how you divide your time between work and social things, and what do you do for social things? And maybe you could say a bit about, this sleeping and eating there.
Okay. So we each have our own room. They're very small, pretty much like a cell in a prison. You got to put them those but we each have our own room. We have 3 meals a day, breakfast, lunch, and dinner. And for the people working nights, we have a big leftovers fridge where we just go and whatever we want. We get cooked meals 6 days a week. Sundays are like a day off from station. But like for me and Carl, our days kind of depends on what happens to the detector. So we don't really have days off, but we're very flexible. Like we want to do some fun things when nothing is happening, that's fine. We don't have a regulated schedule. And then we have things going on. Tonight it's going to be volleyball. We had a Mardi Gras party last Saturday. We're going to have Green this coming weekend. Carlos is running a bit in the gym. I'm rowing. I'm skiing. So we try to keep ourselves busy. But work and recreation kind of go hand in hand. So we're kind of always involved.
Okay. Well, thank you very much, Sven and Carlos.
Applause for all of our scientists. Thank you.
Bye.
Hey, yeah.
I believe that the general rule is at 30 below, with a 30-mile-an-hour wind, bare flesh begins to freeze in 30 seconds.
Yeah, I would comment, I have not been there during the winter, but when I was there during the summer, and the temperatures were often between 20 and 50 below centigrade, you couldn't go outside with any exposed flesh, but the equipment they gave you was really, really good, and it was not bad going out. I mentioned the experiment was kind of a kilometer from the South Pole. It was really not too bad to walk it.
Thank you, Spencer. Thank you, lab scientists. So we're going to begin the public Q&A, and again, there are microphones here, and at the top for those who are in the balcony. But before we begin, I just have a couple of questions. So Tamas, a point of clarification. I wasn't sure what lignocellulose depolymerization was, so it was an of course. So can you explain it for me?
Is an eloquent way to say to break down cellulose, lignocellulose. Plant cell wall has evolved to be a very complex, very sturdy structure. And believe it or not, people are studying plant cell walls over 100 years. And if you Google it or try to find on the internet, is there a model how these components, lignin, hemicellulose, cellulose, and some other polymers, are mixed together and how they look like actually in the cell wall? You will have a hard time.
So useful for biofuels, perhaps?
Ultimately, yes. What we like to do is break it down. Remember, there's a 1 billion ton challenge just in the US. 1 billion tons of agricultural waste are available every year, annually, for biodegradation, sugar formation, which then can be converted to biofuel.
Okay.
But what you need is good enzymes to break down, depolymerize.
Okay, I have it now. Thank you.
Thank you.
For the group, so what is the sort of joint attraction here for extreme science? I mean, is there something called calm science that other people do? What's the unifying principle? I mean, you could have chosen to do other things, but you're going to these environments and studying what you do. Any connection there? Any reason why you're doing it in particular? No?
That's where the interesting stuff happens.
Yeah.
Yeah.
Okay.
I mean, whether it's in microbes or materials, too. Right. I mean, that's where the...
Yeah. We know there are these enormous accelerators somewhere in the universe, and we just don't know where they are. And even though after 100 years of effort, literally, because cosmic rays were discovered 100 years ago, and just it's amazing trying to find them or figure out where they are.
So Caroline, I know you have something to say here.
Yeah, I was going to say, for me, it's just also this kind of thing where you're just impressed. We think we're humans, we've got big brains, we're really advanced, but here are these microbes that can survive things that we can't even dip our finger in. So I think and there are many more of them than there are of us, by mass or by number. choose your.
Should we be fearful of that?
No, no, but they're essential for keeping the planet running. They're essential for cycling carbon, for cycling nitrogen, for cycling iron, and that's what keeps the planet alive. But you also see the amazing things that they can do, and that's really exciting to me as a scientist.
Tamas, the same attraction for you?
I'd be more modest and say two things. One is, if you go out to your backyard and grab a handful of dirt, you have the same chance to find some interesting organisms in there than near a volcano. But how was that with the Himalaya? Why did you climb it, Mount Everest? Because it's there. So if you have a chance to go to extreme environments, it's more likely that you will find organisms which we have not seen before, capabilities which we have not seen before. So it makes perfect sense. But I said I'll be more modest. In my book, basically any environment is extreme in one way or another. Because it doesn't have to be an exploded nuclear power plant in order to find unique organisms. Look at it, how many unique organisms have been found just on the human body or in the human body, simply because the tools are so much better today than 20 years ago. So.
Okay, one last question from me, and then I'll leave it to the audience. But what does this tell us about the possibility of finding different kind of life forms on other planets? There seems to be this bias towards carbon-based organisms. So does this give us any insight into what we might find in some other locations? Caroline, you must have an
Go
answer.
ahead.
Go ahead.
Can I yield to your wisdom?
It's only speculation, but two things. One is, if we try to find life as we know it, there are fair chances that we will miss it. Because if you look at the periodic table, there's not much reason why life decided to become carbon-based on our planet, while carbon is not among the 10 most abundant elements of the Earth's crust. And there's so much more silica. And if you look at the periodic table, silica behaves pretty similar to carbon. So if you try to look for life as we know is here on the planet, there's a fair chance we'll miss it. But on the other hand, there are certain things, and astrophysicists will tell us how we can approach that, which will prove that there is biology ongoing. The smart biologists state that where there's water, there's life. And where there's life, there are microorganisms. So I will have a job.
Okay. All right, with that, let's start. Do we have someone at this mic? Yes, right here.
Yes, hi. A question maybe for Dr. Torok and maybe the others. Recently in a soil study class, I think the characterization was made that even in the backyard, as you describe, something like only 5% of the organisms are thought to have been characterized. I'm not sure what that means, but perhaps you can talk about that, and you offered to talk perhaps about the detection mechanisms that we have now that we didn't have before. You said both cultured and other types of ways to characterize or find living forms. If you could help us.
Thank you very much for the question. Microbiology dates back to 1670s when Anthony van Leeuwenhoek as a first human being, saw what he called little animals. He was the first one who opened up a new world which humankind didn't even know about it. It would be the same value as somebody tomorrow would find life outside the planet, something totally new. But in the past history of microbiology, microbiologists usually cultivated or tried to cultivate organisms. And it is a very difficult situation because microorganisms hardly ever live alone in the environment. They always live in communities, and the communities are very strongly organized, regulated, and interdependent on each other capabilities. Now, if you pull out organisms out of that environment, put them into the laboratory where you use nutrients which these organisms in nature have probably never seen, and try to grow them as a pure culture because that's what traditional microbiology wants and the industry needs. You need an organism if you want to make antibiotics, so that there's logic behind it. But ultimately, that leads to the now understanding that since we cannot simulate the environment and the conditions in the habitat where the organisms was isolated from, it is very difficult to grow organisms. And non-culture-based techniques, which take the environmental sample and characterize something in it, like extract all the nucleic acids and try to figure out how many different organisms live in that environment, have shown us that depending on what organisms we are talking about, but in case of bacteria, we probably have studied less than 1% of the organisms living in nature. In case of fungi, the number will go up to maybe 5%, but the young generation has still plenty opportunities to discover new organisms, because for 99+% of the microorganisms, we know very little what they look like, what they actually do, what is their role in the environment. But there are very good techniques and the development of technologies which can help microbiology to move on are exponentially getting better.
Thank you for the question. This gentleman here.
Dr. Torok, you showed us pictures of genetically developed insecticides, wherein a crystal is eaten by the bug and the bug dies because his intestines are torn up. What happens if I eat it?
The insecticides are natural, these biological insecticidal molecules are natural products, so they are not genetically engineered. The genetic engineering is when we try to put that capability into the plant, so that the plant will grow up and produce its own proteins, which will then destroy the insects. These are pretty harmless for us. They are only specific to the insect, actually so much more specific that if you want to kill certain insects that are typical for a certain plant in a certain habitat, you better isolate the organisms, the toxin-producing organisms from that very environment, because that microorganism and the insect, they have been interacting with each other for a longer time. You cannot just simply take any organisms from anywhere and produce some silver bullet which will kill any kind of insect. But I'm not aware of any scientific evidence that these so-called delta endotoxins, which are insecticidal, would be harmful to humans.
Have they actually been eaten by humans?
At the moment, most of the time, when you see the crop duster over the Central Valley, it's most likely that they are spraying Bacillus thuringiensis, one of these bacteria which is producing these endotoxins. So if you don't wash your fruit, vegetable, you might. But there are many other crop plants already which contain BT capabilities, Bacillus thuringiensis capability, BT corn, BT some other crops. And I'm not aware of any scientific evidence that there would be any harm. Otherwise, FDA would not allow, or the USDA would not allow their use.
Thank you for your question. I think we have some up there. I can't really, yes, here?
My question kind of expands on what you've been talking about. The nature of farming is to change your environment, kind of make your own ecosystem to better yourself. And I'm really excited to see developments in science that help that end a little more naturally. But then also, to what end should it stop? Being a recent addition to the Berkeley area, I was immediately surprised at how many people really cared about having natural food, organically grown, locally grown, stuff like that. Do you know how this sort of research will end up impacting ecosystems as a whole, or kind of locally to the farm? Or is that something that will just be considered, I guess, later on down the line?
Caroline, do you want to comment on that?
So I think one of the things you have to realize is that if you look at the Earth as a whole, it's an environment that we as humans have radically altered. So even, as you say, even farming itself is a radical change. But basically, we as scientists are aware that, and the public is aware, that genes confer new traits. And so we're very interested and very cautious about trying to make sure that whatever new traits we give to, for example, food-bearing plants, we try and build in as many safeguards as possible to prevent the spread of those traits to other natural organisms. So we frequently not only build in one, but several of those, so that basically those organisms need, for example, specific nutrients from us in order to survive. So that they can't just sort of live free and go wherever they want.
Thank you for your question.
Thank you.
Let's see. Do we have someone over here? Yes.
Yes, and this is for Spencer Klein. When you were detecting the neutrinos, how did you know they were neutrinos and not something else?
Well, for the most of them, as I said, we're looking for things that were going upward through our detector, And we don't know of any other particle that could go all the way through the Earth and then interact below the detector and go upward.
Okay. That fits, okay. And then for Andrew Miner, question for you, when you were working with magnesium metals, well, being from active duty experiences as well, I realized that that catches fire, there's nothing that's going to put it out. What can you do ahead of time to mitigate the particular problem?
Yeah, that's right of, really small little magnesium particles and things like that. But a big bar of magnesium, it has a passivating oxide on it, so it's completely stable. So when it's in a big form that you would build onto a car or something, it's not ignitable like that.
Okay.
Thank you. Someone here?
So I know that the ice cube detector is buried under hundreds of feet of ice to prevent cosmic radiation from setting it off. So how does Ariana stop that?
Well, that's a good question. I should say, even though Ice Cube is down 1,500 meters, there's still a lot of cosmic ray particles going downward through it. There's about 500,000 times as many particles going downward from cosmic rays, interactions above it, as there are from neutrinos going upward. And that's why we have all of these phototubes to look at the pattern of the light. Ariana uses a different technique. It's only sensitive to very high energy neutrinos. So it will only detect cases where there's a lot of energy deposited in a very, very small volume. And that's something you would pretty much only get from a neutrino interaction, an ultra-high energy neutrino interaction. In addition to that, the downward going cosmic rays, you would develop a shower of particles in the air as it goes down to the detector. So our plan is to probably put occasional antennas on the detectors sticking up into the air to look to see if there's anything coming down.
Thank you. We have one more upstairs.
In a time of starvation, could we use the genetic code of these microorganisms in extreme places to modify humans to not use as much water as they do now?
I don't think I heard the question.
Could you repeat the question? We had a little difficulty hearing it.
If in a time of starvation, could we use the genetic code of these microorganisms to change humans to not use as much water as they do now?
That's a great question. That's a good question. I'm glad I don't have to answer that. I'm just
You want to come to work for us?
Yeah.
Let me put it that way. The first time when I meet my new class every semester, the first thing what I teach them in biology, you never say never. Because tomorrow, somebody will show up with something that nobody has seen it before, and then you have egg on your face. So I don't know the answer, but I would never say never.
I don't think we have anything planned like that.
Yeah.
No,
Yeah.
certainly not.
Anyone else want to add anything? No. Caroline, yes?
I was just going to say, in terms of, one of the reasons I think I'm here before you is because it's really important. Science progresses, and we learn more about what we're able to do. But it's really important for society to understand what we're increasingly capable of doing and decide what we can do and what we should do. I don't think any of us here make those decisions. It's really the world as a whole, the society in general, that really needs to make those decisions. We try and learn we're capable of doing.
Okay, we have another one over here. Yes.
Hi, thanks for the talks. I was just wondering if the microbiologist had any idea exactly what the fungi were sensing when they're moving towards the radiation source, like the radiotropic behavior? Like what they're actually detecting, or if they're just moving toward heat or something like that?
Let's put it that way, that we are only so far that we observe this capability. and colleagues in Ukraine and here in the United States are working on these experiments to repeat it under laboratory settings. And about a handful of publications are out there, which are really interesting, but we don't know what the mechanism is. What's interesting that most of these organisms are pigmented, black pigmented organisms, very rich in melanin. And melanin is known to be a kind of a oxygen radical scavenger. So it could be some role there. There must be a reason why most of these organisms are so deeply black pigmented that can survive and show this positive radiotropism feature. About 25% of the fungi which we collected have shown this capability, and most of them were collected very close to the exploded nuclear power plant, either directly on the block by robots, collected by robots, or nearby. We don't know the mechanism yet.
Thank you. Next question.
What has prevented the evolution of macroorganisms that live in boiling water or subacid lakes?
What is preventing?
That's it. You want to?
No, go ahead.
Okay. So one of the things that microbes have evolved to do is to be able to get energy from many different sources. And however, when basically the amount of energy you can get by reacting, for example, sugar with a metal, moving the electrons, is actually much, much smaller than what you can get from moving electrons from sugar to oxygen. And so that's one of the reasons that you find larger organisms usually rely on oxygen, because they can just basically get more energy from the chemical reactions that you can do with oxygen. We'll see what happens. Evolution is not standing still.
In other words, the organisms would not have enough spare energy. Every cell needs to produce energy. You don't have enough energy for differentiation.
Yeah, exactly. I mean, I think it's just sort of macroorganisms need a lot more energy for all the little specialized functions that we have, whereas microbes, they're pretty simple, and so they can grow and divide with a bit less energy.
Well, is it also that most of the world is, not 7 pH? I mean, so most of the evolution that's happened, it happens in not such extreme environments?
Most of the
I mean, so you got these lakes that are very acidic, and these microorganisms are evolving there, but most of the world is not so acidic.
Right. So you definitely have more evolution occurring in non-extreme environments. That's correct.
Actually, organisms which live under extreme pH, extreme environmental conditions, whether very acidic or very alkaline, they evolved to have mechanisms to differentiate between outside pH, which is so extreme, and the internal pH, because the internal pH in those cells is still very close to the neutral. Otherwise, the machinery wouldn't work. But they had to evolve capabilities, which are, some of them are passive, some of them are active, energy requiring, introduction requiring mechanisms to spiff up the cells and the protection of the cells so that they can withstand and make a living under those extreme environmental conditions. Because there's a reason why we use bleach and clean up stuff, because that would kill microorganisms. And the same for acids. But there are organisms which live way beyond that bleach pH or the very acidic pH. But inside the cell is still close to neutral.
Thank you. I think we have another question from the balcony.
Hi. Hello. So I found what you presented, I think, Tamas, on the Chernobyl and the fungi there, very provocative. And I know there's been a number of different studies I have looked at of the life around the radiation region in Chernobyl. And frankly, for the last I don't know, 50 or more years, you've had a very real stigma against radiation due to a lot of things, linear no threshold theory, etc. A lot of these things have been disproven. So one, I wanted to ask you, well, because most people just don't take it into account that that not all radiation is bad, what have we learned of the benefits of that radiation could possibly have for human beings? Because, and what are the limits that we have so far of knowing what role this actually does play in life on Earth, the evolution of life and mankind?
Ladies and gentlemen.
Thank you very much for the question, but I don't think I can answer that for several reasons. One is because, as I said, microbiology and its applications are very much technology dependent. We need the tools, and the tools are getting better and better and better, which means that we learn a lot more every day. Have we learned how we can benefit from radiation? I probably am not the one to answer that question. At Berkeley Lab, we have scientists who are studying longer-term space travel, where cosmic radiation is an issue for shielding the astronauts and the people, and what are the potential damages that longer-term space travel may cause. We are at the beginning. The fact itself that we can discover that organisms can survive, that in high radioactivity containers, high radioactivity waste containers where the pH is tremendously alkaline, the temperature is high, close to 100 or 100 degrees C or above, and we have incredible radiation, microorganisms live and actually require that. The fact itself that we can discover that is already an incredible step. The next logical step would be, you're right, how can we benefit? It's not all bad, but I wouldn't be the person to answer that at the moment.
So is radiation necessary for life?
Certainly contributes to evolution. We have background radiation constantly bombarding the DNA, all of our DNA here on the planet, and that has contributed to evolution. There's no question about it. Survival of the fittest.
Thank you.
Thanks.
There is another question over here. Yes.
Hi. Once more, thank you very much to LBL and to the scientists here for this program in general and the really exciting things that you've presented tonight. Thank you very much. Dr. Miner, you talked about a phenomenon of smaller objects being stronger than larger ones, and that linear relationship that you described. I'm not familiar with that. Is that entirely due to surface tension, essentially, in those materials, or is there some other way that you can describe or characterize why that occurs?
Yeah, it's a very good question. I mean, in these structures that I showed, one of the reasons it occurs is because those defects, when they run through the material and they hit a surface, they escape, and there's no more defect. And so materials become more and more sort of perfect as you deform them, and that's one of the strengthening mechanisms. But on the other hand, there's other mechanisms at these small scales, like the ability to create new defects from the surface. It's actually easier to create these dislocations at a surface than it is inside. And so oftentimes there's a lot of competition between dislocations escaping and dislocations starting, and how they do so and determines a lot of the properties. Also, there's a similar phenomenon in real materials, bulk materials, where you change an internal dimension that I didn't really talk about. The internal dimension can be something like grain size. So crystals, all the atoms are lined up, but inside a real material, you have different grains that all have different orientations. And as you change the grain size, you dramatically alter the properties of the material. So changing these internal dimensions and the external dimensions, these are the handles that you have to play around with the properties when you talk about mechanical properties.
Thank you. Next question, please.
Hi. There it goes. I had a question for Mr. Torok. Do you think the fungi could evolve resistance against the gene built to keep them away, as has happened before with other things?
Why would they want to do that? Textbooks tell us that fungal organisms need organic material as carbon energy source. So there must be some truth to it, although I don't trust really textbooks. But that's another story. No, I don't think they want to be away, because fungal organisms in the environment are the ultimate biodegraders. They can biodegrade all those hard to biodegrade materials slowly, but they do. So I don't think they want to evolve to be away from the plant material, which is their ultimate carbon energy source. And there's not much competition long term for fungi, because either other organisms cannot deal with it, or simply are not as efficient as fungi. So again, never say never, but I don't think there would be any logic in it. Sorry.
I was referring to when you said you'd put the gene into, such as corn plants, and then they would be resistant to fungi growing on them. Do you think the fungi could grow on them and
could overcome that?
Develop that ability?
Ultimately, absolutely, yes. I'm sure we have to keep doing it. So what we try to do is, fungal organisms, like any any organism, is producing some small natural molecules which help them in competition for the little food there is out there for themselves. We call them antibiotics or antimicrobial compounds. So basically, we are looking for small peptides which play that role, produced by fungus X, and has been shown to be successful against currently known plant pathogenic fungi. But since evolution doesn't stop, there will be always new plant pathogens, and there will be always new molecules needed to fight them off. That's how I would answer
Thank you for your question. I believe another question here. Yes, please.
I'm curious about how it's possible for microorganisms to exist in superheated environments. As I understand it, when a protein is exposed to high temperatures, it causes it to denature. The protein unfolds and then links to other proteins around it. What is it that's happening at the molecular level that prevents this from happening in these microorganisms?
Thank you for the question. It's a very good question, because as we know, water boils around 100 degrees C, and proteins start denaturing depending on the type of organisms at much lower temperatures, and the same is for nucleic acids, which also start denaturing around 80-some degrees, and depends again. I don't know the answer, but what I do know is that organisms which live in very high temperature environments, they have additional molecules available, biocatalytic molecules available, which can repair destroyed or broken up genetic material much more efficiently than organisms which don't live under those conditions. For DNA molecules to be available for replication or expression, certain enzymes and DNA binding proteins and many other factors have to be present so that the DNA molecule relaxes and opens up and makes itself available. Just the opposite was found in all high temperature living archaeal organisms called reverse gyrase, a molecule which constantly compresses back, supercoils back the DNA molecule and protects it from. There are specific protein molecules which help with this protection. Plus, as we know, organisms, when they are really exposed to stress conditions, they will produce a lot of brand new protein molecules, heat shock proteins, which again play protective roles. So I don't have a silver bullet answer to your questions, but I know of many discovered and reported scientific evidence pieces which show that these organisms can make a living there.
Thank you. Oh, I'm sorry.
I can chime in a little bit. So it's pretty amazing. These proteins carry out many different functions, and you'll find proteins that carry out functions in, sort of our cells or bacteria that exist at sort of normal temperatures, and you'll find similar, proteins that perform the same function in these very extreme organisms. And what you find is those proteins have been optimized to work at high temperatures. In fact, if you take them down to low temperatures, they don't work at all. So, and they don't denature at those high temperatures. So they have actually adapted to be stable at much higher temperatures. That's actually great for biotechnology. That's one of the ways that we've found detergents that work at much higher in warm water versus in cold water. So, so it's actually been, so we know a little bit about how these proteins have adapted to survive in high temperatures. Do you know?
Just one more comment that so far the highest temperature where organisms surviving and living and actually growing were reported is 121 degrees C. And their enzymes are optimal at 125 degrees C. And there are calculations depending on what the nutritional requirements of the organisms are that upper level of life, upper temperature level of life can even be somewhat higher. It could go all the way probably to 140, 142 degrees C. It blows our mind, yes. You're absolutely right. But that's the beauty of it.
Thank you. I think there's another question from the balcony.
I don't know if you covered this in your speech or not. However, can you tell me how and where are neutrinos produced?
Okay, so neutrinos are produced in a couple of places. One of them is radioactive decay. Well, actually, the main place is radioactive decay. So like in nuclear reactors or in fusion reactions or in the sun, or in higher energies, like at accelerators, they're produced in decays of pions and kaons. So what we think happens is we know there are somewhere these enormous accelerators that accelerate protons and other nuclei to higher energies. There must be some residual gas in these accelerators or residual light. So occasionally the protons will collide with this residual gas or beam gas and produce a bunch of pions and kaons. And when those pions and kaons decay, then we'll get neutrinos. And those are the neutrinos we expect to see.
Okay, thank you.
Thank you. We have time for a few more questions, and I think right here.
I don't. No, there's not been conclusive evidence that there has ever been life from outside of this planet found on Earth. Yes, that is correct.
Thanks. Okay.
Thanks. Another question for Dr. Klein. How often do you find a neutrino, and why are you looking for them? Like, what are they telling you?
Okay. So
because it's there.
They have to distinguish. IceCube currently detects about 70,000 neutrinos per year. But most of those are neutrinos that are produced, for example, when a cosmic ray hits the North Pole, produces this shower of pions and kaons and neutrinos, and some of those will go through the Earth and interact at the South Pole. And that is useful. It's interesting for studying neutrinos. But the real purpose of IceCube is to find these extraterrestrial sources. And what we'd really like to know is, where are the giant accelerators in the universe? I've mentioned these cosmic rays that we've seen. We've seen cosmic rays with a single particle with energy up to 3 × 10⁻20 electron volts, or roughly the same energy as a well-hit tennis ball. And so somewhere in the universe, there's an enormous accelerator. But the problem is the protons, the particles that we observe, are electrically charged, so they get bent in transit. So even if we look at record their direction when they arrive at Earth, we can't project back and see where they came from. And we'd really like to know, where are these accelerators? What are they? How do you get such enormous energies out?
Thanks.
How many cosmic rays? The charged particles, cosmic rays?
The neutrinos.
Neutrinos that are not from the
neutrinos you're looking for.
Well, so far we have not seen any that we have confirmed are extraterrestrial. I mean,
okay, next question.
Well, I should say, IceCube is a new experiment. We've only completed construction about a year ago. We're really just starting to analyze the data. And if we had seen something already, that might have told us we could have gotten by with a smaller, cheaper detector.
You don't want to say.
Now the next question. Yes.
This question is for Caroline, talking about the metal-eating microbes and how, I guess I wanted to hear more about how those would produce biofuels. I just wasn't quite sure.
Thank you. That's a good question. So basically, as Tamas already told you, microbes need a couple things to grow. So they basically need an energy, many things, but the key, most crucial ones are a source of carbon and a source of energy and also a source of reducing equivalents. So biofuels are basically reduced carbon. They're carbons bonded to lots of hydrogens, whereas CO2 is carbons bonded to lots of oxygen. And basically the difference in what the carbon is bonded to is the difference between something that's waste product from a biofuel, CO2, and something that's a biofuel. So what we're really hoping is that basically we can inject electricity into the bacteria as a source of reducing equivalents and CO2 and have the bacteria essentially as part of its metabolism, take that electricity and create carbon-carbon bonds that would form a biofuel. And the benefit of all of that is basically we can harness, we can actually use many different sources of electricity, as well as we can capture energy, solar energy, and convert it into electricity much more efficiently than plants can capture solar energy and create biomass. So basically, we can basically get to biofuels, hopefully, much more efficiently than by going through biomass.
Thank you, and I think you are going to be our last question for the evening.
Okay, I just had a quick question for Spencer. You mentioned that neutrinos are constantly passing through things and that we observe some that interact. Mm-hmm. Do we know why they are interacting so randomly, or is it just chance?
It's essentially just chance. I mean, the higher energy neutrinos are more likely to interact, but it's just occasionally one of them will hit a proton or a neutron in a certain way and react.
Thanks. Spencer, when is your next trip to the South Pole?
I'm hoping to go back to the Ariana site in November.
Okay, great.
But that remains to be confirmed.
Okay, that concludes the program for this evening. Thank you, audience. Thank you, lab scientists. We'll see you again on April 23rd at Berkeley Rep, the smaller stage next door. Thank you very much for coming.